Why virus? Why virus? I mean, first thing we think about virus is, ugh, dangerous. Dangerous if it's the human virus, but the point is that viruses are extremely specific. And across the years, I guess the ultimate demonstration is not a scientist in front saying it's safe. I'm a scientist here and I'll tell you it's safe, go and use it. Are you going to? Probably not. Partly because the scientific aura has been shattered in the last 10 years because scientists from the companies will talk and scientists from the biopesticide will talk, scientists for sustainability will talk, scientists for ultimate resource utilization will talk, so you don't know. So I would say at the end of the day, it's reality, it's the field, and that's why you from the field are probably the best place to judge. So I'll take you to the field and I'll tell you that for years now, developing countries have been using insect viruses because it's easy. You can do it yourself. You can do it in your kitchen. You can do it in your living room or in your hut, for that matter, in your little house with very few. And you grind larvae that you know are dead at the end of the season, and you keep that mixture in a bottle, very often in a Coke bottle that is better utilized for that than thrown on the beach. And you mix it with water the next season and you spray your field. You will say, most of our regulatory agents in the world will say, and what if there are contaminants in there? And they are right. But most of the time they are not. And it's a very efficient method and, as you know, cheap. Why developing countries, why did it start like that hundreds of years ago? It's simply because there was nothing else, or they could not afford the what we call more advanced conventional methods. Yeah.
When you're going to use a Coke bottle, do you just rinse it out with water, or do you use soap first?
When you mix the larvae?
Or if you're going to use like a container, like a Coke bottle, do you have to wash it out with soap first, or can you just do a triple rinse with water?
You finish your Coke and you put it in. The viruses we're talking about, and that is my leeway into what the viruses are, because if I tell you right away what the viruses are, I know what's going to happen to your eyes. You're going to glaze. So if I tell you, oh, yeah.
What should be contaminants? We're talking about that they're very easy to prepare even at home.
Yeah.
What should be contaminants?
They will always be. Insects have contaminants in their guts the same way as we have contaminants in our guts. That's no big deal. The contaminants that are of worried are if, for example, the person who prepares it has listeria, so human pathogens, on their hands. So all they have to do is wash their hands before they prepare the cocktail. Don't sneeze into the bottle or into the mixture when you prepare it, because then you will introduce a human pathogen. Those are the ones we don't want. There will always be pathogens, but that's life. Our body is used to that. But why aren't the viruses themselves a problem? Well, we are sure now because they have been used all over the place and people have been tasting it, and actually Ima and I have been drinking it in front of farmers in Madagascar, because with good reason, trusting the whites is a foolish thing, and I'm sure you will agree with that. So you got to go an extra stretch to demonstrate you're really sincere. Once the trust is established, then things are reversed, and then you have confidence. But back to the baculovirus. Why the baculovirus? Here, hundreds of years of use without, any negative impacts on humans, but not only not on humans, but on mammals. not on mice, although people might want an effect on rats, for example. But no, not on mammals, not on orders that are outside of the arthropod world. And most of the time, not even outside of family. So the GV, for example, one of the baculovirus we're talking about, they are divided. It's not just the baculoviruses, but we have those who are called NPVs, and we have those who are called GVs. So the NPVs are bigger if you look at them with a microscope, and that's what you'll look at later. The GVs are about 100 times smaller, very, very small particles, constantly moving, ectic particles, moving with the van der Waals force of physics. Both are so far from other viruses, especially those that attack mammals, that they have never been seen. And then you can go into attacking humans. And you can go into the genetic of it, and it has been done in the last 20 years. And the way it works will never recognize the DNA pattern that a mammal has. So very specific. For the big industry, not good. They like to sell to a farmer total bug elimination when it comes to a pest. Not so with biopesticide. You have to know which species you have. So it asks for a change in mind, and that change in mind for the growers has happened. So that problem 10 years ago of trying to convince growers of using a specific pesticide was enormous. They didn't even want to hear about it. They wanted to spray even without the pest. That was automatic. Every 15th of June, I spray Lorsban on my field. That was good practice. So to come and say, well, no, you don't spray necessarily on June 15th, and moreover, you don't spray if you don't have that pest, or because that pesticide will attack only that pest. So way easier nowadays, but at the time, that was a paradigm shift. Could I show something on the slides just rapidly? I don't want to lose.
Where do you have it, Chuck?
On the desk. I think it was put on the desktop.
Right now on the desk? Okay. Do you have it in frogs? Is that what you want?
I don't know what was transferred. Frogs? Yeah, it is in frogs as well. Yeah. Just the Kwantlen 2012. Do you have any question until now? So easy to produce, safe.
I have a comment about, it seems like the public perception though, when you say, like if I say to my neighbor, I work on insect virus, they go, ooh, virus?
Mm-hmm.
Are you okay?
Yeah. Yeah.
So how do you get it through to the public that a virus?
It's easier than you think, but something has to be done. Imagine you're in Madagascar. We didn't have to imagine it, we were there. AIDS had just arrived. So, and the culprit were the whites, right? We came with AIDS, and AIDS was proliferating in Madagascar. Now, a couple of years later, the Swiss government says, we have a project, we're going to produce viruses to treat your rice fields.
We're going to spray it all over the island. We'll come with big planes and we'll spray virus all over the island. We just brought in truckloads full of condoms to protect you from virus, but now we're going to spray the whole island. Can you imagine what the government said? We're not particularly in favor of the idea.
We still have our heads, so we must have done something right. What did we do? Village-oriented films and TV-oriented films, where you simply explain in layman terms, but honest, of course, what are insect viruses. And I can guarantee you, that's our experience. Everybody in the village where we worked understood way quicker than the minister. Way quicker.
Yeah, it's true. You know what might also work well, or we made the experience works well, when you tell people, you know, yogurt is made from bacteria, cheese is made from bacteria. Just because it's a virus doesn't mean it's bad. So we look for the good stuff. People eat cheese, people eat yogurt, they are not afraid of it at all, and
Maybe it's a good idea to write something in like the local paper or something.
absolutely.
Absolutely, that's what I meant.
Yes.
Communication
Yeah.
and information is the
Yeah.
key to everything. You open, you re-establish trust. Everybody understand that.
I almost think it's harder in this area than like in the villages in Madagascar, because I find people here think they're more educated than they are, and they
Yeah, but
they're sure they know what's
it,
right.
I know, but it seems that the human part prevails there. We didn't see differences here. We did a film here. It's called a Commando of the Infinitely Small, and you have it where you see production system in Nicaragua, production system in Quebec, what we did, and production system with Martin Andermatt, which is one of the biggest biopesticide producer in Europe, and education, communication, and people, common sense with the people works very well. I mean, you can carry it too far, like the Ontario Prime Minister a few years ago.
I think there's another difference. I think there's also the fact that people in Madagascar didn't have a choice, right? For them it was the question of, am I going to do something about those bugs that eat my crop, or am I not? And so they really had to engage in thinking. While what we found in North America was that people kind of wanted to be told what to do, and the one who speaks the loudest is heard the loudest, right? So we found that there was a lot more willingness to assume responsibility for taking their own decisions than we have found here.
Could you give me the first slide, please? Because, okay, so those are the NPVs that you'll see later. They are a little bit more triangular than the traditional NPV, but that's how it looks like. Next one. We have, those are the baculovirus, if you want. Some will disagree with the first one, the nudie virus, but essentially it's a baculovirus that forgot to get its protective capsule, but it's the DNA. So what are they? They are DNA circular supercoiled thing. And they code, they will recognize, it will work only in certain insect cells. As I said, specific to the family, more to the family when it comes to GV, more to the order, so a little bit bigger when it comes to the NPV. And it's the same with the tissue. Some will attack only the intestines, and some will attack a little bit the intestines and will go into the fat body. Those are very often, ecologically speaking, the best ones because they don't kill the insect, they just use the fat body, which is not a vital organ, to create a mass of viruses without killing the insect, and like there's more production per larva and then more survival in the environment. So yes, there are some in Diptera, there are some in Hymenoptera. They are called different. They have their own baculovirus group, the alpha, the gamma, but it's not important at this point. So you find them in Diptera, so in flies, in Hymenoptera, wasps, a special type of wasp that looks like a Lepidopteran larvae, for that matter. GVs in Lepidopteran, and a little bit in shrimps. And that's a little bit, if you are on the coast of Thailand, you might want to check what you are spraying in your fields to see if there is cross-contamination. NPV is the biggest group, and you have two groups in there which are again based on things that are not so important now. It's the protein that can attach to the cell. How are those proteins? But they all have something like 31 genes that are common. All the rest varies. Those that can attack other tissues, like the fat cells on top of the intestinal tract, they are a little bit bigger because it needs more information to attack a different cell than a single cell. If you can move on. So, just the general view of how all that is happening. The virus is liberated, comes from the larva that dies, is absorbed by a living larva. That's supposed to be the villi in the intestinal tract. By the way, there is a membrane here that is supposed to protect those cells, and the virus will have to find a way through. Young larvae, it's easier; older larvae, tougher. And some viruses have developed proteins to trick the peritrophic membrane, and there are other ways which comes in the formulation, maybe sometimes, of the product to help the particle to reach the cell. Once it's in, makes its way through, does some multiplication, and instead of producing the same thing that is in the larva, it produces a nucleocapsid without the polyhedrin or the granulin, the protective coat, the matrix around it, because it will stay in the larvae for a while. So why create a superstructure that is not needed, take more time, more energy? Let's go right away with the nucleocapsid that are the infective particles and infect other cells. And then only then, at the end, start producing virus particles that will have the protective polyhedrin or granulin protection. Next one. So here's just rapidly how the villi looks like and how the crystal breaks in the gut lumen, goes through that peritrophic membrane through different mechanism. Either it's it takes a fracture in the peritrophic membrane, especially in young larvae. It's not completely done, or it has a protein that tricks the peritrophic membrane, goes in, and then I showed you earlier what happens in those cells of the gut lumen. Can go. Oh, but yeah. Characteristic of insect guts, it's not like ours. Ours is acid, so for insects it's alkaline. It's way up in the 8 and 9, and that's what the virus needs to melt. That protein here will melt in alkaline fluids. Okay, thank you. Very rapidly, it's a detail of how the budded virus are produced, so those that will not be encoded by that crystalline matrix. So it comes in, will be read, and will be produced in the nucleus by a virogenic stroma, where the DNA of the virus takes over and controls the DNA of the cell and tells it from now on, here's what you produce. It goes out and it produces two things. It produces the virion itself, the nucleocapsid, but it also produces proteins that, through an RNA polymerase that the virus has, which is another characteristic of baculovirus, and then will produce those little proteins that will meet the virion as it buds out of the cell. And it is these little proteins here that allows the virion, which now is free in the hemolymph, can go to the next cell and say, oh, it's me, hi, just because of that recognition pattern. And the cells is fooled into believing, oh, that's a friend, come on in. Okay. So back to that whole system, just to wrap up everything. The cells in the lumen, infected, produces budded virion that are not encapsulated. Here is a GV as opposed to the NPV. You saw the NPV, it was kind of icosahedral. You see the angles of the particle. You don't have that in GV. In GV, it's kind of a rounded but way smaller structure and releases the virions. Okay, I think that's it. Yep. So all that machine is specific, more or less specific depending on the virus. There are NPVs that go across families. GVs most of the time stay within a family. That sounds strange, but that's how it is. Question at this point?
When we were discussing or debating over the two methods of pest control, something came up with Matthew here, and he said, would viruses mutate like bacteria? So if you have large numbers, is there a probability that one of them somehow mutates that would then become a potential human pathogen or something that is not beneficial? Is that possible?
They absolutely mutate. They absolutely mutate, and within 2 or 3 generations, they will adapt to the insect species that, for example, if they by chance, and it happens all the time in the field, so you've been produced in Spodoptera exigua, and then you have another Spodoptera or another Lepidoptera completely different, and they are ingested by that larva. Happens all the time. If they want to survive, they have to adapt. And it's not that the individual particle will adapt, that doesn't work like that, but there are so many produced in one larvae that one might have the tweak that will allow it to infect the gut lumen. So that type of adaptation is possible. The other one, I mean, can you have a mouse that will mutate enough to give birth to an elephant? I don't think so, nor vice versa, although the birth will be easier. I'm sorry.
We have 3 kids.
I don't, that's too far. That's the point. Too
Yes.
far, way too far.
Yeah, actually, that's kind of what I said, very unlikely, because I haven't looked into insect viruses, but I've looked at phages a lot, and I mean, that's one of the bonuses about phages is as the bacteria adapt, the virus adapts to the
Yeah, that's it, within bounds, but not,
within bounds. Yeah, yeah.
that's another world.
I mean,
Yeah.
I have a question on this topic. What do you think came first, the virus or the insect?
Well, the virus, a virus is a dead thing. It needs life.
Yeah.
So, but the virus first didn't have an insect. There was no insect. So in the history of thing, there was first, I don't know, a pre-shrimp. Or before the pre-shrimp, there was an amalgam of cells. There was an hydra, maybe. So first, that's where, but as soon as you have life, you have somebody to profit from life. We know that, it's life, right? You're here because you have a creator seed in you. Well, all creators know that for every creator, there are 10, oh, I want to be diplomatic here, help me with an English word that would be appropriate. No, well, those are the negative, but those that profit from profiter, or they depend from you, opportunist, or yeah, that and they count on you. And it is not bad in itself. It is when the profiter kills, not necessarily murder, but you know, eliminate, put to the side, ridiculize the creator, that the problem comes. But we're when you're creator, there will be somebody that comes. So the same with virus and bacteria. That's how there is something to gain from something, so it appears. That's nature. Opportunity, opportunity, opportunity.
But so then that answers your question, right? So now, what was there first, virus or insect?
Insect, according to what Jacques is saying.
Yeah. But that doesn't mean that there were insects before there were viruses, right?
Exactly.
Just in this
it's a complex
specific
answer.
combination in insect and its virus.
To get you going.
But viruses are dead. There's no doubt about that. Viruses are dead. Only life brings it to life. That's.
I think there was another
life brings
question.
life.
So I'm just imagining the Coke bottles full of stuff. So after the virus has left its host and is encapsulated, how long does it remain?
Storage. If you let the larvae died properly, that means you have had not only encapsulation with the polyhedrin or the granulin, but also the envelope. which seals the granule or the polyester. Then comes the question of what's going to eat it away. If it's in a basic fluid, not long. If it is...
Jacques, here is the slide, right? Here's the naked virus. And here are the viruses when they are encapsulated.
Yeah. So if they are like that, and if they are at 7 or 5 in terms of pH, and if they are at room temperature, about a year. If they are in the fridge at 0, 2 years. If they are at -20, 50 years. Acidic, they tolerate up to 4 approximately. So from 4 to 8, you're fine for a good while. Now, of course, if you have contamination by a bacteria that produces alkaline juices, and it's not in the fridge, it'll be shorter.
When we look into real life, you know, get out of the lab situation and look into real life, the way we did it in Madagascar was the growers just stored the virus in the insects the way they were, and left them at room temperature in any kind of container, an old can, whatever they had, and let it sit there until the next growing season. So they collected the insects after harvest, infected them then, let them die, and let them sit there until the next growing season. And only then did they squeeze up what they had left and treated their fields with that.
Good way not to have contamination. Dryness doesn't invite fungus and bacteria.